Hello everyone. In this session, we will look at cytoskeleton. And this is going to be a very brief introduction to cytoskeleton. Now, just as a kind of simile, okay, or analogy, let us understand the fact that as human beings, we are able to stand erect. We have a shape, okay, where you have a distinct organization of every part of the body, and we are able to move voluntary. Actions are possible by us because of the skeleton, and also we are able to brace against any kind of external force because of the internal skeleton that we have. Therefore, the skeleton is not only giving us a kind of structure, it is also enabling us to do functions other than a structure or a support. So, similarly, every cell in our body has a cytoskeleton. That means every cell has a skeleton of its own, and that skeleton is able to give the cell a structure, the cell an organization, and the ability to carry out several functions. So, this is what we are going to look at in this session. So, let us look at the learning outcomes of the session. The cytoskeleton is a complex, dynamic network. When we say dynamic, it means that it is an ever-changing network. Once it is formed, it is just not present as that. You have a building being formed. A building will remain so for years together. So our skeleton once formed, our skeleton will remain as such for years together. But within the cell, it is not necessary that the cytoskeleton always remains the way it is. It will assemble and disassemble, and that is the reason why you are calling it as dynamic. That means it is constantly changing. And this network is of interlinking protein filaments that are present in the cytoplasm of all cells, right from the nucleus to the plasma membrane. So it is extended, it traverses the entire cytoplasm of the cells. There are three cytoskeletal components, that is the microtubules, intermediate filaments, and the actin filaments, which are also called as microfilaments. The cytoskeleton are associated with a remarkable phenomenon of dynamic instability, as just mentioned. That is, it has the capacity to assemble, and it also has the capacity at the same time to disassemble. So it is kind of the assembly and the disassembly, or the polymerization and the depolymerization of these cytoskeleton is something that is going on constantly within the cell, and that is what is understood by dynamic instability. Apart from being a structural support for the cell, the cytoskeleton contributes to several functions of the cell, such as cell motility, cell division, signal transduction, etc. So now let us look at one by one the different functions or different aspects of the cytoskeleton. As you can see in this fluorescent stained plant cells, one is very clear that you can make out that all the plant cells are cuboidal in shape. So a certain shape that is present for a particular cell type is attributed to the cytoskeleton. So you can see how these cytoskeleton that are stained are able to form a cuboidal structure. Also, what is very clear is that the entire cytoskeleton is traversing the entire length and breadth of the cell. It's not that it is present only in one part, but all the three cytoskeleton together are able to form a very delicate and yet very strong meshwork or network of the cytoskeleton. In fact, even this in the nucleus also is positioned, and it is present in a particular area in a particular structure thanks to the cytoskeleton itself. So therefore, we can say that the cytoskeleton organizes the structures and the activities of the cell at the same time. So, having understood that a cell has all the three cytoskeleton, let us look at which are the three principal components of the cytoskeleton. One is what is called as the actin filaments, or it is also called as microfilaments. As you can see, the layer just below the plasma membrane or the membrane is definitely microfilaments, and it is not just restricted to this portion, but majorly it is present at this portion to form what is called as the cortex region. And this, as you can see, is supporting the membrane. So the membrane does not collapse because you have the cytoskeleton supporting it. Secondly, that it is ovoid in structure. That means the cell is ovoidal in structure is also because the way the cytoskeleton is ovoidal in nature. So the structure or the architecture of the cell type is dependent on the structure and architecture of the cytoskeleton itself. Secondly, what you observe is that the second principal component is what is called as microtubules. And again, you can see that microtubules are present all across the cell, originating from close to the nucleus and going right till the plasma membrane. So it is traversing the entire membrane. Plus, you have the third component, which is the intermediate filaments. Again, with respect to intermediate filaments, you can see that it is traversing across the entire cell. And in fact, even the shape of the nucleus per se is determined by the intermediate filament because it is giving the spherical structure to the nucleus from outside as well as from inside of the nucleus. So that is again something that we are kind of reiterating again, that is, it organizes the structure of the cell. Now let us look at what are the activities of the cell that the cytoskeleton contributes to. Before that, let us have a glimpse of the, not in detail but in brief, the principal components of the cytoskeleton. So like mentioned, there are three components: microtubules, actin filaments, which are also called as microfilaments, and intermediate filaments. What is important to note is that microtubules are the largest cytoskeleton component at 25 nanometers of diameter, and the structure of the microtubule is such that it is a hollow cylinder. So inside you don't have anything, it is hollow, but you have this entire thing as a cylindrical structure and it is very strong a structure. So therefore, it is able to help brace external stress or external force when it is present inside the cell. Second, it also acts as tracks, or it also acts as, just like you have railway tracks, similarly microtubules can act as tracks. And it can also hold on, or it can also space out different organelles within the cell. We will look at it in a minute. Then the second component is the actin filaments. The actin filaments are the thinnest filaments at 7 nanometer diameter, and each filament is made up of two polypeptide chains, and these two polypeptide chains are coiled around each other. So they have a typical coil cable structure. You know, you can see the electrical cables in which you have two wires which are coiled around each other. Same is the structure of the actin filaments per se. Then you have the third component, which is the intermediate filament. The name itself is suggesting that its size is intermediate between the microtubules and actin filaments. So at 10 to 11 nanometer diameter, it is placed between the microtubules and the actin filaments. But these are cylindrical structures, but they are solid filled. They are not hollow like the microtubules are. So they are solid filled filaments. And again, when you have several of these intermediate filaments together, they become a very strong composite structure. One another important fact about these three cytoskeleton components is the kind of proteins that constitute these. So microtubules are made up of a specialized type of proteins which are called as tubulins, while actin filaments, as the word suggests, are made up of actin molecules, and intermediate filaments, unlike microtubules and actin filaments, are made up of an array of proteins. It is not just one type of protein, but several different types of proteins make up the intermediate filaments. And so intermediate filaments, you have several different types as well. So this is something that one has to keep in mind. Now let us look at the different functions of the cytoskeleton. To begin with, let's understand the fact that you have these cytoskeleton actually enabling positioning of organelles within a cell. So for example, you have the endoplasmic reticulum positioned here. You have the mitochondria positioned over here. You have the membrane getting a structure because of the microfilaments that is there. So the microfilaments kind of forms the base for the or that becomes a support for the plasma membrane per se. So these cytoskeleton therefore can act as scaffold, and they're not just maintaining the cell shape. So you can see that it has a cuboidal shape, that is thanks to the microfilaments, the cytoskeleton per se, and the structure as well. And plus, what is understood is that the positioning of the nucleus, the positioning of the endoplasmic reticulum, the positioning of the Golgi, the positioning of the mitochondria, everything is because of the way the internal network of the cytoskeleton is, and they hold these organelles in position. Now we come to another function of the of the cytoskeleton, and that is that it is enabling what is called as intracellular movement of various cargo proteins or of various organelles as well. So these are what is called as the microtubules. So what you see over here are the microtubules, and they act as railway tracks. Why are we calling it railway tracks? Because on these you can have molecules moving that is able to carry molecules from one part of the cell to another part of the cell. So for example, if you want something to move from the nucleus over here to the membrane, then it is on these microtubules, or the by using these microtubules, that you have the components being transferred from the nucleus to the cell membrane. Or you can have it the other way around, from the cell membrane to the endoplasmic reticulum, from the endoplasmic reticulum to mitochondria. So these movements of molecules or components present within the cytoplasm is thanks to the microtubules. So you can get a glimpse of how that happens. So intracellular movement of materials and organelles is because of that. So therefore it is important to understand that these are kind of functioning as tracks on which these organelles can be moving. So this is a molecule which is carrying an organelle, which is carrying a protein that moves from this part of the microtubule to this part of the microtubule. You can see how it is going. So therefore it functions as such. Now what is also important to know is that cell locomotion or motility. So we just saw intracellular motion, but we can also have cell locomotion happening. So for example, you take into consideration the fact that you take into consideration the fact that I think I will show you the movement per se. Okay, so you can see, before I go into cell motility, so you can see how on the tracks you have the molecules moving. You see, this is how you have within the cell organelles the molecules moving. Now let us come to other types of locomotion or motility that is associated with the cytoskeleton. So for here example, let us take as microfilaments, and we all know that muscles are made up of actin and myosin. Okay, and actin and myosin together are responsible for muscle movement. So you can very well see over here, okay, that you have this as the sarcomere. So this is entire unit of one actin line to another actin line, and you can see how there is contraction, sliding of the myosin with respect to the actin, and therefore you have contraction and relaxation happening. Just have a look at it again. Okay, so you can very well clearly see how the contraction and and the relaxation is enabling muscle movement to take place. Then you have, for example, this over here is the cilia, which is present on the surface of the cells, moving rhythmically in one pattern. And so effectively what the cilia can do is that the surrounding environment that is present, that can be propelled thanks to the movement of the cilia per se. Okay, so this is again nothing but cytoskeleton. So the cilia is made up of microtubules, and so the movement of the cilia is thanks to the microtubules, which is nothing but cytoskeleton. Similarly, what you can observe over here is, say bacteria have certain flagella, and these flagella enable the bacteria to propel itself. So the movement of the entire cell is because of the bacteria. But you must understand that similarly in our system, you have the sperms also moving in a similar fashion, where you have its tail flapping to have the entire sperm cells move. So therefore, again, this is an important fact, and it allows, or the microtubules allow the entire cell to move. So what has to be taken into note is that you just do not have only intracellular movement, but you can also have the cell being motile, the cell moving from one place to the other place, thanks to the cytoskeleton again. Now we go to what we understand by the fact that cytoskeleton also plays a role in cell division. So here you can very well see how within the cell per se during the mitosis phase, this what you see is exactly at the point of metaphase, where you can see all the spindle fibers are holding the chromosomes at the equatorial plate. So the formation of these spindle fibers are key to enabling the chromosomes to be aligned at the equatorial plate during the metaphase. And the formation of the spindle fibers is because of the microtubules. So spindle fibers is nothing but microtubules. So when the cell is in interphase, that is, it is not in the mitotic stage, you will never find spindle fibers. It is only during cell division that you will find the spindle fibers being formed. So here is one indication that it is not necessary that a cytoskeleton is present all the time in a particular cell. So when the cell is not dividing, you don't have the spindle fibers. But when the cell is going to divide, you have suddenly the spindle fibers being formed. So here what is there is, once the spindle fiber is formed, you have the spindle fibers again getting deformed, because in a cell that is not dividing, you will not see the spindle fibers. So this is a complete indication of the fact that the cytoskeleton can assemble and disassemble. So one has to keep, therefore, the phenomenon in mind for cytoskeleton, and that is that of dynamic instability, where you would have the assembly and disassembly of the cytoskeleton happen. Here again, what you can see is very clearly that you have cytokinesis. So during cytokinesis, what you understand is that you would have slowly, gradually, the microfilaments forming an involution over here, and then suddenly you will find that these two cells are forming two cytokines. Now, therefore, let us look at this video. Okay.
Mitosis begins with prophase.
So, yeah,
Notice
please
the
listen
DNA
to it.
condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
Right. So you must have understood how, you have the cytoskeleton ensuring that at the required stage you have the spindle fibers being formed. At the required stage you have the spindle fibers retracting back, and then you have the cell dividing at the cytokinesis that is happening because of actin filaments. So you can see how dynamic instability of the cytoskeleton is enabling cell division to happen. This is something that is very, very interesting and important with respect to the cytoskeleton per se. Now, just continuing with the fact that the cytoskeleton can assemble and disassemble, just giving a view of another video so you can see how actin comes together to form the actin filament. So these are all actin molecules, and these actin molecules are polymerizing to form the actin filament. But another important factor that you should understand is it began polymerizing over here, and you can see that the actin is slowly extending at this end, and at the other end it is depolymerizing. So you have polymerization at this end happening, and depolymerization at this end being this happening at the same time, by lieu of which you have the actin that originated here move on to here. So you can see how, due to the assembly and disassembly of the cytoskeleton, you have the cytoskeleton moving inside the cell, and in lieu of that you will have the cell moving. So if you understand endocytosis, okay, endocytosis is a process where you have the a cell giving rise to pseudopodia. So how do you think the pseudopodia is formed? It is thanks to the actin filament actually extending, and that extension is leading to what is called as the formation of pseudopodia. Another interesting function that is associated with the cytoskeleton is with respect to the fact that it can act as a signal transducer. Now, what do we really mean by that? So, for example, consider you have a surface molecule. So, this is a membrane protein and this is a transmembrane protein called as cadherins. So, like all transmembrane proteins, you have the membrane spanning region, you have the intracellular domain, and you have the extracellular domain. So, you can see how neighboring cells, so this is one cell and this is the second cell, how the neighboring cells are interacting with each other. So there is cell-cell interaction, but recognition of the cell-cell interaction is happening because you have the intracellular domain of the cadherins attached through two different proteins to actin filament. Okay, and this actin filament changes its conformation when the cadherin on the outside is binding to this cadherin. So that change in conformation of the actin is going to induce other changes inside the cell to give rise to a physiological response, and therefore cytoskeleton also can act as signal transducers. So with this, we kind of have understood what are the different functions in which the cytoskeleton is involved with respect to a particular cell or with respect to cells. So let us make the conclusions. Cytoskeleton comprise proteins that make microtubules, microfilaments, and intermediate filaments, which all together create a network that ensures that the cell does not collapse and it has a particular structure. Their role in intracellular as well as cellular motility is significant. The movement of the cilia and flagella is due to the cytoskeleton. Cytoskeleton plays a role at every stage of the cell division, with the dynamic instability of the cytoskeleton helping in moving from one stage to another stage. As cytoskeleton present intracellular are associated with several membrane proteins, they are able to mediate inside out as well as outside in signaling. That means they are able to carry out a signal transduction. So the skeleton of the cell, cytoskeleton, enables the cell to maintain its shape, its integrity, its movements, its multiplication, its communications, its interactions with the surrounding environment, etc. Absolutely dynamic. Thank you.